I guess what I'd like to achieve today is to provide you with some baseline information on microbial ecology, what we know from the science of that field, and have you think about some of the information I present so that you can actually look at some of the claims that are being made from biological product manufacturers and see for yourself whether perhaps the claims stack up against the science that I'm going to present you with. There's a lot we don't know at the moment, so as a discipline group we're still learning a lot about what goes on in the soil, and you'll find out what some of the challenges are in a little while. So some of the things I want to cover today is the question about what microbes live in the soil, and I'll probably touch on the bigger things besides the microbes. So I'll talk about the micro, meso and macrofauna. I'll make mention of those. I'll also talk about what they do, and when you know about what they do, we can actually put a value on those microbes. I want to also talk about what regulates their activity. We're talking about living organisms, and like us, they are influenced by their environment, their habitat. So I want to talk about some of the conditions under which they operate and what they have to put up with. And I'll just touch upon some of the commercial products and services in terms of some of the claims. So I don't know who, did anyone see this program on the BBC about Life on Us? Because I think it serves a big, yes, it's a fantastic program, and it serves to illustrate the important role that microbes play on the human body. So to start with, I want to perhaps run through a list of features that are used to describe the soil communities. So we use taxonomy. For a long time we probably got hung up on taxonomy, but it's now just one criterion that we use. Another way of looking at the microbial life is to group them according to their size, and we've done this for a long time. So we have groups like microflora, which I'm going to spend most of my time talking about this morning. The viruses that are here, the bacteria and the fungi, and we've got size ranges there. Fungi tend to be the biggest and viruses the smallest. They've got the microfauna, which are the protozoa and the nematodes, the mesofauna, which are collembola and mites, and the macrofauna. So we're starting to get more, these are starting to be more visible to the naked eye. And we use these as sometimes as surrogates. If we've got lots of earthworms, we assume that below that trophic level there's also a lot of other smaller invisible organisms operating as well. So if we take a gram of soil, what have we got in it? So we've got, estimates have put it between 10,000 and 15,000 species. We've got those species are very highly adapted to that environment. So whatever is in that environment chemically, you'll have a community there that will be adapted to that. As I mentioned, technology has come a long way. We've, in the environmental sciences, we've piggybacked a lot on the medical area, and they've been working on the development of tools for a long time. So basically, in a short period of time, we've gone from looking at things on agar plates, so artificially developed agar, growth media, through to extracting DNA directly from the soil on site and then assessing the DNA, so decoding its DNA and having a look at what genes are present, which informs us about what that organism does in that system. Very powerful technology. So we've gone from having an understanding of about 1% or less of the community that exists in the soil to we're edging up towards a 40% or more understanding of actually what's there. So the point I want to make here is that in the last, say, 5 to 10 years, we've come a long way in discovering what's in the soil. So we're in discovery mode in a lot of ways, and we're still trying to come to terms with how the whole system works. So the other thing that we look at is where microbes or where biota occur in the soil environment, and generally they occur around their food source. So they tend to be located in the surface soils, and most of them are there. You will find things deeper. They're located around roots. If you were to pull roots up, you see that rhizosheet of soil that clings to the roots, and that's clinging because the roots are producing exudates and the soil's clinging to that, and then you've got a lot of microbial activity going on there, and sometimes you'll see earthworms hanging off those collars, and they're hanging off from there because they're grazing on the bacteria and fungi that are living there as well. I want to just switch now to why they're important, why we've got to use them. And you can list a whole range of very important functions that we rely on the planet so we actually can get by and survive. There's some really important functions. So they cycle carbon. They do that by photosynthesis. Most people probably don't regard microbes as being photosynthesizers. We see them as being mainly decomposers, but they can use CO2 and convert that to organic forms of carbon. They regulate plant nutrient supply. There's lots of things they convert. Nitrogen, phosphorus, potassium, they sequester iron through a whole range of different interactions. Part of that means they capture and release greenhouse gases. They produce and use methane. They produce nitrous oxide and carbon dioxide. They improve soil structure. They produce gummy substances, and just by virtue of their hyphal networks, they actually improve the aggregate stability. They degrade pesticides. This is a free service. We take this for granted. Whenever something's biodegradable, it's biodegradable because there's biology degrading it, but there's limits to how much they can degrade. They regulate water quality by acting as a filter. They prevent pesticides from getting into waterways by degrading them, and they suppress soil-borne diseases. Most of our antibiotics come from the soil, from soil organisms, particularly belonging to a group that's dominant, which is the actinobacteria. And I don't know whether you've been reading the papers over the Christmas period, but there's been a revival in the search for new antibiotics from soil environments. Some work by Chris Franco in Flinders University has been searching for these, screening for these new antibiotic products. They come from the soil environment, and they're an ecological strategy developed by soil organisms to keep competitors at bay. And as you know, disease is established because there's an imbalance, and so they grow in number, whatever the disease organism is, and basically start to push into the niche of other organisms. And if those other organisms can produce antibiotics, they can keep them away. And they're a resource for valuable genetic material. I just want to mention the fixation of nitrogen and the free living fixation of nitrogen. And some recent work by Gupta has estimated the contribution of free living nitrogen fixers. So free living, they live freely in the soil, as the name suggests, but they're different from symbiotic nitrogen fixers, which form the nodule association linkage. So I'm not talking about those, I'm talking about a whole lot of other organisms that live freely and fix nitrogen. And it's been shown, this is in cropping soils in the southern region, so we can show between 0.2 and 3 kilograms of nitrogen per hectare per day can be fixed. Now that's a big range, but there's good reasons for why there's such a big range, and it's to do with factors such as clay content, water, and a few other biotic or abiotic factors. But we can control this a little bit by feeding the nitrogen fixers. They need a lot of carbon. They need 10 grams of carbon to fix 1 gram of nitrogen. So they tend to prefer wheat cereal, wheat stubble to cereal rye to canola, so they have preferences for their carbon source. We find that if you have summer grasses, such as Rhodes grass and panicum species, they can actually harbour not free-living nitrogen fixes, so over summer you can actually build up your reserves of organic nitrogen. And fertilisers actually inhibit or reduce or suppress these fixes. So I just want to mention that microbes also influence the phosphorus cycle. So we've heard of vesicular bustial mycorrhizae. I just want to make mention of those, and they help certain host plants assimilate phosphorus. I'm going to skip over that one. And the genetic resource, this is some figures from a chemical company manual, which shows that the number of entries of new products that are biopesticides, and there's an expectation that market will grow significantly in a little while. And these are just some of the products that are emerging as potential biopesticides, so fungi, bacterial extracts, which are extracts of bacteria, and bacteriophage, all of which have been registered as potential new products. So what regulates their activities? There's primary and secondary regulators. Some of the primary regulators we can't really do that much about. We don't water, temperature, clay percent, pH, and then there's secondary things that we perhaps can manage a little bit better. The quality of the organic matter is also a consideration, the C to N ratio. The higher the C to N ratio, the more you will find a tie-up that the microbes will pull the nitrogen back into their cells and not have it released. So the general rule, so all of these things have various C to N ratios, and the general rule of thumb is that anything with a C to N ratio of 25 to 1 or greater will stimulate tie-up of nitrogen by the microbial community, because they get first dibs at it. They're much better competitors than plants are for that nitrogen. If you've got a low C to N ratio, so if you fiddle around with your residues, you will stimulate mineralisation or release of the nitrogen back so that plants can take it up. Finally, we're looking at microbial products, and there's lots and lots of them on the market. They're getting more and more, prevalent in the marketplace as people are looking for alternatives to the more traditional forms of fertilisers. What we tend to see is that the package that's presented goes like this. You have scientific terminology to define the problem, which is fairly robust because there's a lot of information on the science of a problem. We often get the commercial test to verify the problem, so that's good. We then find that there's always a product to fix the problem, or there might be a product to fix the problem. And then we have either anecdotal evidence or a commercial test to verify that the improvements have occurred. So this is usually how it rolls out. Just a word on tests. There are lots of tests available, but not that many available commercially. But if you look in the literature, there's quite a long list of things that you can actually measure. I've highlighted the ones here that are becoming, or that are commercially available. I guess the point is that claims on how products work and their success in terms of changing the community or the functions in that community haven't caught up yet. So there's still work to be done in terms of the development of appropriate tests and validation of those tests, because how many of us have experienced putting a test in, getting the results back, getting a number and not really understanding what that number means. And part of the development of the test means that you need to understand this range of variation, and so you need to be able to explain whether the value you've obtained sits within the normal range of variation, or whether it's actually, you know, a valid, a robust measure. And the other thing that needs to happen with that measure is an appropriate management recommendation. And often we don't see that. So I'm getting to the end. I think it's the last slide, or might be the second last slide. So I just want to finish by saying I've painted a picture of what the community is like in the soil. And so hopefully you're getting a sense that introducing microbes that are grown in a luxuriant soup in a bioreactor in some building may not be able to work very well in the soil environment. We might have to do certain things to that product. So last one, buyer beware. There's lots of new products available, but I think you've got to ask yourself a few basic questions. Does the product work? What's the evidence to show that it works? Have the claimed beneficial effects been validated in the field? Are there trials, you know, like these ones that are run for rhizobial inoculants? Is the product safe? You know, does it contain organisms that might be risky to human health? Does the product specify what it's got in it? Is there any QA that it actually contains what it says it does? And does the product identify the conditions under which it performs best? When we're talking about living organisms, they don't perform well everywhere. So, you know, what we want to hear is someone say, yeah, the product works good under these conditions, but doesn't work so good there. If they say it works good everywhere, then I'll start to raise the eyebrows. And I will leave it there, so thanks for your attention.